Hello everyone, I'm Professor Gonzalez-Fernandez at Lehigh University, and today we are talking about this new publication from my lab called Versatile Cell-Penetrating Peptide for Multimodal CRISPR Gene Editing in Primary Stem Cells. This publication was recently accepted in the journal Advanced Functional Materials with an impact factor of 19. And I have with me today a grad student in my lab, Josh Graham, that was the first author of the publication, and he's going to tell us a little bit about what he did and the results that we got. So first of all, I want to do a short introduction about the challenges that we were trying to address and the main objective of this research. This research was focused around CRISPR delivery. So the main bottleneck for the application of the therapeutic application of CRISPR therapies is the delivery of the CRISPR machinery. So the delivery of CRISPR with high efficiency, especially in primary cells, like for example mesenchymal stromal cells, which are the cells that we use in this study, is very challenging. And it's very challenging because we need to get the CRISPR machinery across the cell membrane and sometimes even into the cell nucleus. And by doing that, we can damage the cell membrane and cause high cytotoxicity. So usually CRISPR gene editing and the delivery of the CRISPR machinery is performed through viral methods. But these viral methods, they are challenged or they are limited because they have high toxicity and activation of the immune system. For example, last year, a patient that was in a CRISPR clinical trial for CRISPR therapy for the treatment of Duchenne dystrophy in the muscle passed away sadly due to a huge immunological reaction against the virus that was used to deliver the CRISPR machinery. So another option for the delivery of the CRISPR machinery is non-viral. So we can use different non-viral methods. For example, we can use electroporation or different nanoparticles like lipid nanoparticles, lipofectamine, and polycationic methods. For example, polyethylamine or PEI. These are safer than the viral counterparts because viruses can insert into the DNA randomly, and that can cause some problems, for example, the activation of oncogenes. And also viral vectors, they usually activate or produce the CRISPR machinery in a constitutive way. So that means that the CRISPR machinery is produced constantly, and that increases the chances of the Cas9 nuclease to cut into a different off-target sequence and produce some off-target effects. In comparison to that, non-viral vectors, they just deliver the CRISPR cargoes into the cell, and these CRISPR cargoes that are usually mRNA, plasmid DNA, or ribonucleoprotein, they just get degraded inside the cell, so the action is very transient. But non-viral vectors, they still have some limitations. So one main limitation of non-viral vectors, especially for primary cells and primary stem cells, is cytotoxicity. So non-viral vectors, for example, lipid nanoparticles or lipofectamine, they are highly cytotoxic and they can hinder the capacity of the stem cells to self-renew, proliferate, and also differentiate into different lineages. This is a very big problem because if we are using these vectors for the CRISPR delivery into the cells, we can definitely or completely hinder their stemness and their capacity to regenerate or have a therapeutic action in vivo. Also, another limitation of these vectors is the lack of versatility. So CRISPR can be delivered into different molecular formats or cargoes. CRISPR can be delivered in plasmid DNA, messenger mRNA, and also into the ribonucleoprotein Cas9 guide RNA complex. But not all the non-viral vectors can deliver all the different CRISPR cargoes. For example, in the case of lipofectamine, we need one type of lipofectamine for plasmid DNA, one type of lipofectamine for mRNA, and another type for the ribonucleoprotein complex. And this lack of versatility limits a lot and negatively impact the biomanufacturing workflows for the production of edited cells. So the main objective of this research was to address these limitations and bottlenecks by the optimization of a new cell-penetrating peptide for the delivery of CRISPR across different CRISPR cargos, and also for different systems of CRISPR gene editing in primary mesenchymal stem cells and also in vivo. So in order to do this, we work with a specific strategy, which is a cell-penetrating peptide. Specifically, we work with the RALA cell-penetrating peptide. RALA stands for arginine, alanine, leucine, alanine, and is a 30 amino acids peptide that was initially discovered by Helen McCarthy's lab in Queen's University Belfast, and I optimized during my PhD for the delivery of plasmid DNA into mesenchymal stromal cells. So again, this cell-penetrating peptide is a 30 amino acid sequence that is positively charged due to the presence of arginines in its sequence. And thanks to this positive charge, the peptide can complex to negatively charged plasmid DNA and also negatively charged messenger RNA. So the cell-penetrating peptide complexes through charge interactions with these different cargos to make different types of nanoparticles that then can be delivered to our cell of interest. Okay, so now I'm going to pass it to Josh, who is going to talk about the results and also the experiments that we performed for these.
All right, so most of the cargos that RALA was optimized to deliver were much smaller than the Cas9 protein and other CRISPR cargos. So our first goal was to make sure that we could effectively encapsulate and deliver CRISPR machinery with the RALA cell-penetrating peptide. So first, we checked the size and charge of our nanoparticles, including pDNA, mRNA, and ribonucleic protein. And we were able to show that all of these nanoparticles fell within the charge range of 20 to 30 millivolts and the size range of a diameter from 100 to 200 nanometers. So since these are effective for endocytosis, we then moved on to cellular transfection, and we showed that we could effectively transfect our MSCs, and then we also compared the transfection profile of RALA to commercial standards polyethylenamine, PEI, and lipofectamine. And in doing so, we showed that the reduced cytotoxicity of the RALA cell-penetrating peptide and the effective transfection rate were able to produce much higher yields of healthy and proliferating cells compared to these standards. So once we had this established, we then sought to dive into gene editing. Our first goal was to perform a CRISPR knock-in, and to do so, we delivered plasmid DNA, 2 plasmids, one encoding the Cas9 protein and a guide RNA targeting the ROSA26 safe harbor locus, and another one that encoded the donor template, which encodes a red fluorescent protein and nanoluciferase gene. And we were able to deliver both of these plasmids into cells, resulting in significant red fluorescent protein and luciferase expression. This also gave us a cell line to assess the knockout rates later on. For gene knockout, we delivered Cas9 ribonucleic protein, and we were able to produce a 70% reduction in this RFP transgene, and which is comparable to other cell-penetrating peptides and other standards for CRISPR delivery. After establishing knock-in and knock-out, we then moved on to CRISPR-A, or CRISPR activation. and we chose to activate therapeutically relevant genes. In this case, for osteogenesis, we looked at BMP2 expression. For chondrogenesis, we looked at TGF beta 3. And then for tendinogenesis, or the formation of new tendon, we chose tennomodulin. And from designing guide RNAs for each of these and optimizing the guide RNA sequence, we were able to achieve roughly 20-fold activation of BMP2, 1.5-fold activation of TGF beta 3, and nearly 100-fold activation of tennomodulin. And interesting, the wide range here is largely due to the difference in basal gene expression in each of these genes. TGF beta 3 was highly expressed in the non-transfected stem cells, so even though we were able to increase the number of transcripts produced, the fold change doesn't reflect that, since it was so highly expressed originally. So at this point, we have achieved the knock-in, knockout, and activation of specific genes in our stem cells, and we wanted to apply RALA to in vivo CRISPR delivery. For this, we chose to target the tibialis anterior muscle right here in mice that constitutively express luciferase and green fluorescent protein, or GFP. So we targeted this insert sequence and then looked at, after 1 week, at the tibialis anterior muscles to look for a decrease in GFP and luciferase signal, and we were able to achieve a significant reduction in both after 1 week, proving that we could effectively deliver the RALA nanoparticles in vivo.
Okay, great. So this is a good summary of our experimental approach and the results. So now let's go to the conclusion here. So give me 1 sec that I delete this, and then we'll go through this last part of the research. Okay, so finally we arrived to the conclusions. First of all, I would like to highlight that the RALA cell-penetrating peptide is the first CRISPR nanoparticle system capable of delivering CRISPR across a wide range of cargo formats, mRNA, plasmid DNA, and ribonucleoprotein protein, for a wide range of different CRISPR strategies: CRISPR knock-in, CRISPR knockout, and CRISPR activation. Also, I would like to highlight that the RALA cell-penetrating peptide was able to maintain cell proliferation and cell renewal in comparison to lipofectamine and PEI, which are well-characterized and commonly used CRISPR delivery nanoparticles. This is really important for mesenchymal stromal cells, as the conservation of their cell proliferation and cell renewal capacity is very important for the therapeutic capacity in vitro and in vivo. Finally, the RALA cell-penetrating peptide was efficient for CRISPR delivery and CRISPR gene editing ex vivo. So we got around 70% of knockout efficiency, which is really high for commonly used non-viral CRISPR delivery methods. And also in vivo, and we were able to significantly reduce green fluorescent protein and luciferase expression in our reporter mouse model after injection in the tibialis anterior muscle. Finally, future research will explore the incorporation of the RALA CRISPR nanoparticles into biomaterial scaffolds for the spatiotemporal control of CRISPR delivery and gene editing. Okay, so we are finished with this. I would like to acknowledge my collaborators and also my funding sources, specifically the American Society for Gene and Cell Therapy and the NSF through the Career Award.
And I would also like to acknowledge the NSF Graduate Research Fellowship Program.
And I hope that you enjoyed the video. See you next time that we publish a paper.